So without further ado, I will hand over to Nicholas.
Hi, thank you. First of all, I just want to start off by thanking the organizers, Sandy and Kristen, for putting on this great event and also putting on the previous events in the past couple of years and for allowing me to talk. So, today I'm going to be presenting the work I've been doing for the majority of my PhD, so the best part of 3 years now. And this is really a big collaborative project between myself, Kristen's lab, Alfredo Quinones-Enosas, everyone just calls him Dr. Q. He's a clinician at Mayo Clinic in Florida, and his lab, and also my PhD supervisor, Marcus Owen. Yeah. So yesterday we saw quite a lot of talks on glioblastoma, so hopefully everyone's somewhat familiar with it. And one of the common themes that we heard in all the talks is that this disease almost always recurs. And one of the reasons this disease almost always recurs is the existence of these cancer stem cells, or in glioma called glioma stem cells, the GSCs in red here. And typically what we see when a patient presents is they respond quite well to initial therapy, which is standard radiotherapy is standard of care for glioblastoma. We see a reduction in the tumor size, but a small number of these cancer stem cells, which are highly resistant to radiotherapy, have actually survived. They're able to then divide and differentiate. And in the case of glioblastoma, this recurrence happens very rapidly in the course of about a handful of months. And so one potential treatment that Dr. Q and his lab have been looking at for a while now is called differentiation therapy. And this is based on the idea that this protein BMP4, if you expose the cancer stem cells to it, can induce differentiation of the stem cells, reducing the fraction of stem cells. And so when we apply radiotherapy, the tumor is more sensitive to treatment. We see a bigger reduction in tumor size, fewer stem cells, and a longer time to recurrence. And in theory, if all the cancer stem cells were removed, then this would be a curative therapy. So in the next couple of slides, I'm going to try and hopefully convince you that I'm not crazy and that BMP4 actually is a potential treatment for glioblastoma. So this first plot just comes from TCGA, and I've taken GBM patients and also low-grade glioma, so it's about 660 patients. And I'm just stratifying based on the median expression of BMP4 into high, which is the green line, and low, which is the red line. And we see that on average, patients with high BMP4 expression are having better survival times. Now, the second figure comes from a very recently published phase 1 clinical trial, the first phase 1 clinical trial of a BMP4 therapy in patients. And so what they did here is they took 15 patients with recurrent glioblastoma. So they'd all previously undergone resection, radiotherapy, temozolomide. The tumor had then recurred, and then they were switched onto this BMP4 therapy. The BMP4 actually, interestingly, was delivered with this convection-enhanced delivery system, which Russ mentioned in his didactic yesterday. And of the 15 patients, this is their trajectories in terms of like relative tumor size. And kind of remarkably, for anyone who's familiar with working with GBM patient data, 2 of the 15 patients appear to show complete response to BMP4 treatment. So this was the size of the tumor at recurrence. And then some 27 months later, we can no longer see the tumor on MRI. And this is the same for this patient here. And then 57 months later, there's actually no sign of the tumor. So this is kind of quite remarkable for anyone who's used to working with this disease. There was also one patient who showed a long-term stable disease, which is also quite rare. And the patients were not necessarily only put on BMP4, but the 2 patients who actually showed complete response were only on BMP4 for the whole course of this time. The one patient who shows stable disease, this is a chemotherapy that's sometimes used in glioblastoma as well. So what we were kind of interested is in developing some mathematical models to simulate this treatment and also simulate some potential clinical trials, which clinicians are interested in carrying out in the future. And really kind of one of the motivating things is this heterogeneity you see in patient response. So this looks almost identical to the image that Kristen shows at the beginning of her talks, where she says every patient deserves their own equation. And we have 15 patients with the same disease, recurrent GBM. They were all put on exactly the same treatment, but we have this just wildly different response. I mean, some of them, it seems that BMP4 is doing nothing. The tumor is just going straight out of control straight away. And in a handful of cases which have been picked out here, they're showing complete response and kind of remarkable response to the treatment. So we developed a mathematical model for this. Schematically, it looks something like this. We have the cancer stem cells in here in red. They divide symmetrically and asymmetrically. We have the non-cancer stem cells here in orange to green, which I'm going to refer to them as progenitor cells throughout the rest of this talk. And the key difference between the progenitor cells and the stem cells is that the progenitor cells have what we call a finite replicative capacity. So they can only undergo a set number of divisions. They then become terminally differentiated, represented by these green cells, and they can no longer divide. Whereas the stem cells are able to keep dividing indefinitely, and that's represented by that self-renewal kind of arrow. We have the BMP4 being delivered, and this is delivered through another cell type of these mesenchymal cells, through these non-virally engineered mesenchymal stem cells. This is kind of a whole other talk, so I'm not really going to get into the details of the delivery mechanism. But basically, there's some BMP4 there, and what the BMP4 is going to do in the model is just affect this parameter PS, and this parameter PS just represents the probability of stem cell self-renewal. So it's going to reduce that, and it's just going to shift everything from the red down to the green. So the clinicians from Dr. Q's lab collected some data. This is from 12 patient-derived glioblastoma cell lines. And what they did is they performed the radiotherapy assay. And this is exactly what Heiko actually explained in some detail in his didactic yesterday. So they grew the cells for 2 days in either stem cell media, which is the red, or exposed to BMP4, which is the green. They then applied radiotherapy at kind of standard doses, 2, 4, 6 grays. And then they count how many and from that you can work out the normalized surviving fraction. And kind of what you see, at least in some ones I'm going to pick out, is that what we would expect to see. So in GBM1A here, this is the control data. The red points are quite high. The surviving fraction is high because the stem cells are resistant. But when we add the BMP4, which is the green circles, the surviving fraction is decreased because we've increased the sensitivity to radiotherapy. So we can fit our model to this data. That's shown by the solid lines. But the kind of question is, so what? Or kind of my talk title had something about virtual clinical trials. So how is this going to be useful for the clinicians and for designing clinical trials? And really the key way is that using this model, we're able to quantify the sensitivity between the different patient cell lines. So you probably have some kind of intuition that this GBM1A patient is more sensitive than this QNS522B patient, where the kind of data points are on top of each other. But what if I kind of asked you this potentially more tricky question, which cell line is more sensitive to BMP4? So it's after the coffee break. I'll see if any people have some energy to participate. So who thinks that GBM1A is more sensitive? Hands up. This is the one on the left. Heiko thinks it. Who thinks that GBM640 is more sensitive? Sandy thinks it. So and we have a few others for GBMs. So this is maybe seems like quite a simple question, but is actually quite tricky to answer. And so I don't know what you did, but maybe in your heads you were doing some kind of calculation like this, where you looked at around 4 grays. You looked at what the surviving fraction for the GBM1A is, what the surviving fraction for GBM40 is. From that, you could calculate some kind of fold change. And you might say that GBM1A has a bigger fold change when you add the BMP4, so this cell line is maybe more sensitive. But I was kind of a bit unfair when I asked the question, in that I didn't really specify what I meant by sensitivity. And the great thing about having a mathematical model is I can be very precise in what I mean by sensitivity. And this is precisely what I mean, is I have a single parameter in the model here, this red psi, which represents sensitivity. And we have this PS parameter, which is the probability of self-renewal. And basically, it just varies from a maximum probability of self-renewal, which in stem cell media is hopefully 100%, to a minimum. Well, it's a probability, so the minimum is 0. And then we have we have this combination of BMP4 and sensitivity, which just shift you from this maximum to this minimum value of self-renewal. Well, the BMP4 level was fixed exactly the same between both these cell lines. So the only thing that explains the difference is this single parameter that we're fitting, psi. And so we can fit this, and actually we find out that Sandy was correct, and GBM640 is more sensitive than GBM1A. It kind of looks like GBM640 is way more sensitive than GBM1A. This is not really the case. It's just because it's a 1/over parameter, so it doesn't scale linearly. And the reason for that is kind of another thing that has been implicit, or I've not really talked about yet, is that the proliferation rate, or the doubling time of the cell line, is really important. And this is something that we also have measured. So here, the sensitivity represents how often the stem cells undergo a differentiation event or a self-renewing event. And so if the stem cells divide roughly on the time scale of once every 2 days, but we see this big difference in surviving fraction, that means almost all of the cell divisions were asymmetric or differentiation events. But if it divides multiple times within 2 days, then actually fewer of the cellular divisions need to be asymmetric to end up with a big fraction of stem cells to non-stem cells at the 2-day time point when radiotherapy was applied. So using this and fitting this model, which is really simple, literally just one parameter, we're able to qualitatively rank each patient based on their sensitivity to BMP4. I think for the purpose of time, I'm going to just kind of skip over this, but they also did this proliferation assay, which is just looking at the effect of the BMP4 on the growth rate alone. And it does some kind of interesting, funky stuff, and I'm happy to talk about it in the break, but I'm just going to skip over it. So then we wanted to look at trying to understand this heterogeneity response a bit better. So what we did was kind of what some of the previous speakers have talked about. We just did a global sensitivity analysis. And so here we're modeling the global sensitivity analysis. The response we're interested in is days gained. So we simulate the model under standard of care. We simulate the model with the BMP4 treatment. We calculate the difference. This is the days gained. And then we want to see which parameters are correlated with the highest days gained. And so this maybe looks like quite a few parameters, or maybe it doesn't, depending on what kind of models you're used to looking at. But a lot of these parameters are actually to do with the whole BMP4 delivery system, and it's not particularly important. What I want to draw your attention to is these kind of two key parameters. The first one here is psi. This is the sensitivity to BMP4. So this is just kind of a nice sanity check. We have a model parameter that is literally sensitivity to treatment, and shockingly, this comes out as the most important parameter for predicting treatment response. But the second one, which is kind of maybe more surprising, or at least was not a priori obvious to us, was the proliferation rate of the cancer stem cells, MS. And so what we wanted to look at was we're interested in performing some new future clinical trials, actually first in dogs. This is something that Russ mentioned in his didactic, is that unfortunately pet dogs are quite susceptible to getting glioblastoma. First in pet dogs, but ultimately more in humans. And so we decided to do kind of a simulation study to see what might happen. And so this is something that some people in Sandy's group and Jacob Scott have called like phase I trials or virtual clinical trials. And here what we're going to do to kind of group the patients is based on just these two parameters that we identified as being highly important for treatment response in terms of days gained. We're going to consider a couple of different patient cohorts. The first one is this green cohort. This is just unstratified. So this is if you just kind of naively sampled from the population, which you might do if you hadn't done any of the modeling that we just talked about. And then the second two are looking at two stratifications where we take the bottom 50% of the parameter range or the top 50% of the parameter range. And so then we simulated a bunch of virtual clinical trials for sampling from these. And this is roughly supposed to simulate an early phase clinical trial where we have around 20 patients and they're undergoing standard of care simulation, which is the red lines, and then BMP4 simulation, which is the green lines. And if we just look at the middle plot, we see the patients simulated in the standard of care and control and standard of care and the BMP4 are always identical. So there's always some effect of BMP4. But the effect is not very large, and kind of what the clinician is interested in. The shaded box means that this trial came back as statistically significant, so a p-value of less than 0.05. So only 2 of these trials came back as statistically significant, so it's pretty unlikely you would observe it, especially as there would be much more heterogeneity in the real patient cohort. But if we carefully select our patients based just on these two parameters and not caring about the other ones, we're actually able to see that the effect size of BMP4 is much larger. So hopefully you can kind of see the green lines are further away from the red lines. And in this case, over half of the clinical trials come back as statistically significant. So this box plot just summarizes it, and I'll finish there and just give a big thanks again to our collaborators, Kristen, Lee from her lab, who's worked a lot on this, Dr. Q, and many of the people in his team, and of course my PhD supervisors. Thank you.
Thank you very much. Any quick questions? Okay.
Thank you. Hey, great study. Very nice talk. Were you able to fit your model to the trajectories of the individual patients in the trial?
No. So the The phase 1 clinical trial that I showed at the beginning, that wasn't completed by our group. So we don't have all the data for that. Also, the thing that we need to fit to the data is this radiotherapy, like we need to perform this radiotherapy assay with the cell line to parameterize the model, like to get that sensitivity parameter. So we would need them to have done that.
It looks like from the figure you showed from the trial paper, you
Yeah.
could potentially abstract the individual data points, right?
Hmm?
You could potentially abstract the individual data points.
Yeah,
from that,
we've
paper.
looked at doing some stuff like that, but yeah, we haven't quite done it yet.
It would be interesting to see which of the model parameters would be global, meaning the same for all patients, and which one do you need to be patient specific
Yeah.
to get these two responders. And then, of course, if any of that would be predictable.
Yeah, it's definitely interesting.
Okay, one more very quick question.
Thank you. Great talk. Have you explored timing between BMP treatments and radiation? And how do you anticipate changing that interval could impact the outcome of what you're measuring?
Yes, we have. So that was something I didn't have time to present, but we have done exactly that, basically looking at optimizing the timing between BMP4 and radiotherapy. So that's something they don't look at in the clinical trial. They actually just only give BMP4, but there's a lot of, all the evidence I presented was to do with the synergy of BMP4 and radiotherapy. And so that is something you can do, and we have done, looking at optimizing the treatment. Basically, one of the things we compared as well was looking at the difference between giving a continuous therapy, a convection enhanced delivery, where you give a continuous low basal dose, or if you give a single bolus dose at the time of resection directly into the tumor bed. And in general, it's usually better to give like a low continuous dose. It is usually the best option.
Okay, thank you again.
Thank you.